Thanks, Kwan. Hi, everyone. It's nice to be here today. I'm going to try the handheld mic so I can turn and everyone can still hear me. Let's see if I can get these going. AV team, could we get the slides going? All right. So I'm really excited to be here today to be talking about genetic ancestry and diversity. It's been really nice to see across the talks from the last 2 days the influence of diversity across a lot of those talks, and also the goals of HCA going forward over the next few years, that diversity is one of those main areas that they're really focusing on. And I think you have probably already seen this figure quite a bit over the last couple of days, but I think it's always nice to kind of set the stage to remind everyone that across genetic studies over the last 10, 20 years, we've really seen a rise in the number of studies, but a lot of these have happened in European ancestry samples, and so they haven't been as diverse as we'd really like them to be. And this has been kind of a reckoning in the genetics community over the last 5 years, and recognizing this and trying to move forward to try and diversify our genetic studies. But this is also true in other biomedical areas as well. And just to make the point that diversity as well as genetic ancestral diversity is actually important, there was this nice study by Luis Barrero's group where they had European ancestry samples as well as African ancestry samples, and they look at the comparison of gene expression in their PBMCs and identified a number of genes that are differentially expressed between these two groups, including a lot of chemokines and cytokines. And they were able to actually correlate these with COVID-19 disease severity, indicating that potentially this is a contributor to variation that we were seeing between populations and could be important for then treatment for these different genetic ancestral groups. And so just to hopefully convince those who might not be convinced yet that diversity is important, I hope this kind of gets across that point. And as I mentioned already, HCA is really making it a strategic goal to really include diversity over the next 5 years across the different human cell atlases. But what's been challenging so far is that we don't actually know what the genetic ancestral diversity is of our atlases. And so this is something that we really focused on over the last 5 years, is trying to identify a method that we can use to annotate genetic ancestral diversity, and then also annotate that across a lot of single cell atlases, including HCA. So that's what I'm going to be telling you about today. And so we know that we can identify genetic ancestral diversity for individuals using genetic variants across the genome. We typically would do this in population genetics using SNP genotype data or whole genome sequencing data, but most of the single cell data that we have in these atlases don't have paired SNP genotype or whole genome sequencing data, which means we have to try and rely on other sources of information. And so we were thinking about then trying to pull genetic information directly from single cell reads. And we can do this pretty reproducibly. And so that's what I'm going to show you here. And so there are definitely challenges that come with single cell data and pulling genetic information from single cell data as compared to our whole genome sequencing data. And part of this is that for whole genome sequencing data, we get good coverage across the whole genome, right? So we have the whole genome covered, and so we can look at genetic variants across that genome. And we have pretty good depth of coverage as well, which allows us then to look at genetic variants across the genome. Whereas with single cell data, as you can see here, we really don't have good coverage across the whole genome. So some variants that we might be able to detect with whole genome sequencing data, we really won't be able to detect as reproducibly with single cell data. We'll just miss them entirely. Other regions, we might have coverage, but not enough read depth, meaning that we might call this variant incorrectly. So in this example here, I know it's a little bit hard to see, but this is a heterozygous variant, meaning that this individual has one of each of the alleles at this location. And for the whole genome sequencing data, we have enough reads of both of those alleles that we would be able to call it as heterozygous. But with the single cell data, we don't have enough depth that we might call it as homozygous. And so this was a challenge that we had to contend with when coming up with a method to annotate ancestry from single cell data directly. Another challenge is that a lot of genetic ancestry estimation methods require you to have the same genetic variants across all samples to look at genetic admixture and PCs, as I'll talk about on the next slide, across all of these samples. And so we knew that this wasn't going to work for single cell data across these atlases, right? Because if you think about even just 5' data versus 3' data, we're sequencing different ends of the gene, right? And so we're never going to get a complete set of variants that are across all of our different samples. And so we needed to find an approach that would allow us to look at genetic ancestry on a sample per sample basis. And so we came up with a method to really help with this. And a few of the things that we were thinking about when trying to develop this method is that we really want it to be scalable. We want to apply it to atlases, right? So we want to apply it to thousands of individuals. So we need it to be a very scalable method. I was also really interested in having it be very reproducible, meaning that when I run it on the same data set that you run it on, it's the same in multiple different labs. But also if I run it on my samples and you run it on your samples and your lab, that we can directly compare the results to one another without having to transfer any raw data. So that was quite important to me. It does come with some caveats, but it allows us to then be able to compare between results pretty easily. And of course, it has to be accurate, right? There's no reason to do this if it's not going to give us reproducible, accurate data. And I'll talk about the admixture and PC estimations in a second. So we came up with this method that we're calling scAncestry. And so what we do is we call genetic variants from the single cell reads themselves. And so this is from 3' data, attack data, SMART-seq data. It doesn't matter. As long as you have sequenced the RNA, then we can call those genetic variants. And to deal with some of those issues where we don't have good coverage for some variants, this next step is really important. It's where we phase and impute variants, and it uses nearby variants to help inform variants that we might not have good coverage for. And so this allows us to then correct some of those variants that we've called incorrectly. It then allows us to identify those variants that aren't don't have good quality, and we can remove those before we estimate genetic admixture. So we have two different methods that we can look at for genetic ancestry. One of them is genetic admixture. And here I'm showing you one individual that we've estimated, and you can see about half of their genome can be attributed to the yellow ancestral region, and then a quarter each to the blue and orange ancestral regions. The limitation here with doing this is that we can't get to a very good resolution, and so we're usually looking at continental level admixture, so European ancestry-like or African ancestry-like. And so in some studies, we'd like to get to more resolution. And so we also built in the ability to look at principal components. And so here what you're looking at, each point is a different sample, and this places individuals that have more similar genetic ancestry near each other on this principal component space. And so this allows us to get to a much more quantitative measure of genetic ancestry as compared to our genetic admixtures. And so to test this out, to see if it's actually accurate, we again went to this data set from Luis Burrows' group. And we use this data set because it has a diverse set of samples from African and European ancestries, but it also has paired whole genome sequencing and single cell data, which means that we can use our gold standard whole genome sequencing data to compare and see if we're actually getting accurate genetic ancestry estimates from scAncestry. And so here I'm showing you the admixture results. And so these are the proportions of these different ancestries for each of these 90 individuals. On the x-axis, you're looking at scAncestry estimates, and on the y-axis, you're looking at the whole genome sequencing estimates. And so you can see that they follow this XY line really well. So there's a very strong correlation between these two estimates, meaning scAncestry is doing a pretty good job here, and we're pretty happy with this. And when we look at then our principal components here in the colored dots, I'm showing you our reference samples. So these are whole genome sequencing samples that allow us to put all of our individual single cell samples or whole genome sequencing samples in perspective. And so each of the black dots are our samples. And so on the left-hand side, you have your whole genome sequencing data. On the right-hand side is scAncestry. And while this isn't a perfect replication, we see that we have a bit of shifting. Overall, we're doing pretty well at placing these samples within this principal component space. And so having validated this, we moved on to try and apply this to as many single cell samples as we could across different single cell atlases. And so here I'm going to show you the results from over 15,000 samples across 4 different atlases you can see listed across the top there. And you can see the distribution of where we were able to obtain these samples from or where they're located online, so you can go and take a look at them. This is from 384 different data sets and covers 21 different technologies. Many of you probably aren't surprised to see that a lot of these samples come from 10x 3' or 5' data. And most of the samples are from primary tissues, so not from cultured cells. And so that's more than 90% of the samples are from primary tissues. Many of these are from blood, so about 3,000 of the samples that we've processed are directly from blood, with about 1,000 from lung, and then good representation from a lot of other tissues as well. And finally, we have about 56% female representation here, so about a pretty good distribution between females and males in the sample from these different atlases.
All right, so now we get into the meat of it. So this is the genetic ancestry results. And so what I'm showing you here is across the x-axis, we have all 15,000 samples. And then on the y-axis, we have the proportion of the genome that can be attributed to different ancestral regions. And I've just binned them by the largest proportion of the genome that can be attributed to one genetic ancestral region. And so what I want you to notice here is that largest group, the first group there that's mostly in blue, those are the individuals that have the highest proportion from European ancestry-like and smaller proportions from other ancestry-like regions. And so we're not getting as much probably diversity as we want to, but this helps us identify regions of the world that we might engage to build diversity into the atlas. And now I'm going to show you the principal component results from these same 15,000 samples, but I'm first going to orient you on this principal component. So on the bottom right-hand corner, these are all of our reference samples. So these are all whole genome sequencing data that we use as a reference to identify where different genetic ancestral regions are from and place our samples on this map. So in the bottom right, we have our European ancestry samples. In the upper right-hand corner, we have our East Asian ancestry samples, and then on the left-hand side, we have our African ancestry samples with other ancestral regions in the middle there. And so when I place on all of these samples, you can see that we really do span across the genetic ancestral space, which was reassuring to us. But when we look at the distribution, then we see that we have a pretty strong, many of our samples are actually in the European ancestry sample region, which is consistent with our admixture results. So we're also interested in where these samples are actually coming from. And so we went through each of these papers and curated where all of the samples were pulled from. And so about a third of them come from North America, with about 20% from Europe and then 10% from Asia, Australia, and South America. And so when we take a look at the genetic admixture and principal components, so the genetic ancestry of the samples from each of these regions, in Asia we see that most of the samples are of East Asian ancestries, and with a small proportion of South Asian ancestry-like. From Europe and Australia, we see that the largest proportion are from European-like ancestries. And when we look at North and South America, we still have a large proportion from European-like ancestries, but a larger proportion from African-like ancestries and Indigenous American-like ancestries, but still a small proportion. And there's not many samples that, in at least the data that we've looked at, that are directly collected from Africa, but 5 of these samples were available in this data set, with 3 of them having a high proportion of African-like ancestries. So now that we've curated all of this data, we're working on a few more analyses, but one of the things that we're now going to be doing is trying to make this accessible for the community. And so we're doing this through a few different methods. One is that we'll have an interactive browser so people can look at, say, a specific tissue type that you want and identify potentially different genetic ancestries and build out a study using this information. We're also working with CZ Cell by Gene and HCA on ways that this data can then be represented in their portals. So CZ Cell by Gene will be including the genetic admixture results as part of their portal, as well as on census. And then HCA, we're in discussions on probably using it for both tracking the diversity that's being included in their atlases, and as well as the distribution of diversity in those atlases. And Blake Bowen, a bioinformatician in Joseph Powell's group, has really helped us really build this into a tool that people can use, and it's finally ready for beta testing. So this is just a picture of the GitHub page, and he has 5 simple commands that he's taken from my very complicated SnakeMag file and made it very easy for people to use, which means that it's 5 commands, and then at the end it gives you genetic ancestry estimates that are consistent with the estimates that we've used, and you can use the exact same reference. And we'll give you nice figures out at the end there, which allows you then to see where your samples are genetically. All right, so looking forward, I'm really excited that HCA has made it a focus to diversify atlases and really focus on this over the next few years. And CZI ancestry networks, I think, are really going to make a big difference as they start getting populated into these different atlases. And our future work will now be using this database effectively to start looking at how ancestry and other biological variables are impacting things like gene expression or attack profiles. So I've gone a little bit longer than I wanted to, but this was a pretty nice collaboration between Joseph Powell's group, Tim O'Connor's group at the University of Maryland, and Shan Prabhakar's group here in Singapore, as well as CZI with Elaine Hsu being my real partner in crime on this, running a lot of analyses and helping me brainstorm a lot of analyses to run. And then thanks to all of our funding sources as well. I'm happy to take any questions and comments, and I'd love to hear how you are thinking about potentially using this and any recommendations you would have for visualizations in the future.
Thank you very much. That's very interesting results.
Thank you. This is a very interesting talk. I'm just curious imputation depends on your panel, right? Heavily depends on your panel. And you estimate only like major components, but within even European ancestry, there are many minor components. And if we talk about the disease-related variants, they're actually not equally distributed among different like subcomponents. How deep you can go with this approach?
Yeah, so I think there are a few different things in there, right? So you're asking about the reference for imputation. Is that part of it?
Yes.
Yeah, so we use 1,000 genomes in HGDP, which are a pretty diverse reference. So that's the reference that I was showing there, right? It has a variety of people from across the world. And I think the benefit of the methods that we used is they're using a lot of variants across the genome, right, for prediction and projection into these spaces. And so you're not relying on one or two variants, which would be like rare disease variants, right? So they won't be relying on those. And they're looking at locations across the genome where we know there are genetic variants. So we're not really finding like somatic variants or rare variants that would be really disease causing. Does that help at all?
Yes. But like if you try to reconstruct your ancestry just based on the variants you called from the data, is it very different from what you do after the imputation, what you get after the imputation?
You mean if we do it before we impute and phase the data?
Yeah, well, if you just within one data set, if you just try to estimate ancestry based on the data, and then you compare it to the imputed data, how far?
It's not very good if you use it without the imputation and the phasing. That was a really big step for us to be able to build that in.
Yes, so for the interest of time, maybe we'll save the question for the break. Yeah, so Mariana gonna introduce our next speaker.
So it is my pleasure to invite the next speaker, Yulia Medvedeva. Sorry, Medvedeva, and she's from Russia, and she's gonna talk about multimodal single-cell atlas.
Okay, thank you. Today, I would like to show you our preliminary results on building a multimodal single-cell atlas of immune dysregulation in T2D in different ancestries. You know that type 2 diabetes is actually a global burden for all countries, and probably in 20-25 years, actually, in many countries, the number of people affected by T2D and pre-diabetes will increase in some places like more than twice. And in Russia, it's also a problem. As you can see here, as you can see here, we have different populations of different ancestries, and the frequency of T2D varies dramatically here. Like in our major population, Russian population, the frequency of pre-diabetes in diabetes is quite high. Also, we have some Asian population, our populations with a major Asian component. And there, the frequency of these diseases is also quite high, but also we have people in the southern part of Russia, they live like close to the mountain. And surprisingly, they have a very low frequency of type 2 diabetes, and it's not only because, well, it's not because their medical system is not good there. It's just something that they do or something who they are, which actually leads to these low numbers. So this is a map of Russia with the overall distribution of people with type 2 diabetes. And as you can see here, on the east part of Russia, the frequency of T2D is quite high. Also, we have quite high accidents like near big cities, especially in Moscow and in Volga region. But here in the mountains, especially in this part of the mountains, it's actually surprisingly low. So we started this project with the idea that physiological properties of T2D is different in different ancestor groups, and we should take this into account when we develop preventive measures or treatment strategies. So we started our project with the small pilot cohort, and then we collected 3 more cohorts in Tatarstan, Bashkiria, Chechnya, and Yakutia. Genetically, these cohorts are quite different. This is a PCA of different populations in Russia. You can see here that our, well, sorry, I think it doesn't really work. Yakuts are like really far to the right. Tatars are on the bottom, and Chechens are on top. And if we look a little bit closer into the admixture, we see that Yakuts and Chechens, they have very specific genetic components which are not typical for any other populations, while Tatars, Bashkirs, and Russians, they are kind of mixed of different components, but the proportions are quite, well, just proportions are right. After all, for our freeze, we are analyzing 3 cohorts, Tatars, Chechens, and Yakuts. They're genetically different. They all have about 30 people with, well, more than 20 people with type 2 diabetes, and more than 50 people, healthy people. So these cohorts are different in terms of anthropometric features and biochemical features. The key observations here that Yakuts, they have really different lipid metabolism. They have high levels of HDLs in healthy people, while in people with T2D, it changes to high level of LPLs. Chechens, they have high BMI, while Tatars, they actually have a relatively low BMI. We actually can see the match between these clinical observations in our lab. For example, in Yakut population, this is population that lives in a very, very cold condition. When we process their blood samples, we actually just physically could see the drops of lipid drops in the serum. And I think this is something that helps them cope with these extremely low temperatures. So when we looked at the single cell data, we saw that surprisingly, when we do statistics, we see significant differences in abundances of all major cell types. But when you look at the UMAP, the difference is not that pronounced. So it was kind of frustrating from the beginning. But then we switched to different projections of this data. For example, this is a reconstruction of regulatory networks done by, based on transcription factor activation, done by Sinek. And here you can see that in almost all the, in almost all on the right UMAP, you can see that in almost all cell types, we see different activated regulons. The light blue ones are specific for T2D, and we see them in all the major T cells like CD4+, CD8+. We see these differences in monocytes, especially in CD14 monocytes, but also in CD16 monocytes, in B cells, and so on. So in all major cell types, we see different activation of transcription factors, transcription regulons. And when we look deeper into these regulons, majority of them are, of course, related to low-grade chronic inflammation. But we also saw some other, like NF-kappa B, but we also saw some other regulons. And when we look deeper into cell-cell interaction network, we saw that the majority of the target genes, they belong to CD14 monocytes. So we decided to focus on these cells for the beginning. We saw that these targets are kind of linked to T2D, so we decided that our primary target would be CD14 monocytes. Of course, CD14 monocytes from people with type 2D, they demonstrated a very heavy signature of inflammation. So this is a clustering based on inflammatory genes. And this red square on top belong to samples with type 2 diabetes. But if we look into the details, it's not like just the number of CD14 monocytes. It's more like some features of these monocytes are kind of different. So this UMAP in the middle represents differential abundance of cells, of CD14 cells in people with type 2 diabetes. So the red dots represent cells which are more commonly found in people with diabetes, and the blue ones represent healthy people. So as you can see, we have kind of a, well, it's not a cluster, but it's a part of the CD14 monocytes, which are more typical for people with T2D. We looked for differential expressed genes between green and blue, and we saw that upregulated clusters in T2D patients belong not only to immune cell activation, but also to oxidative stress. And surprisingly, we saw glucose transporter there as well. And in the healthy cluster, it's also about inflammation, but it's more like antigen presentation, recruitment of immune cell, balancing the cytokines, and so on. And surprisingly, we saw some differences between different ethnicities. It's not that pronounced, and unfortunately, we do not see a very clear link to a particular ethnicity in the enrichment of this area, maybe in Tatars, but also not that clear. Unfortunately, probably due to the sample size or to some technical variation, we do not see clear differentially expressed pathways between these cohorts, but at least we see some differences. And then we performed the subclustering of the monocytes, and we found out that, actually surprisingly for us, that clusters are typical for T2D on the left. The cluster number 0 is actually kind of classical. It's not intermediate monocytes, but it's classical monocytes, but they are expressing much lower of the marker genes. And we can conclude that it's not that CD14 monocytes in T2D are totally different, but they are kind of losing their identity to some extent. Then we looked a little bit deeper into T cells, and we saw similar differences in CD4 naive, CD8 naive, and CD4 central memory cells. The interesting thing we observed that naive cells are the known fact that they are decreasing with age, but in people with T2D, the number of these cells decreases faster. We cannot explain that, but we believe that it's kind of linked to naive T cell exhaustion in T2D. So I'm trying to briefly summarize our observations. We see that CD14 monocytes in T2D, they show kind of reduced lineage identity. They also show strong signatures of adaptation to high glucose level and oxidative stress. age-dependent decrease in T cells, in naive T cells. It's more pronounced in people with type 2D, probably due to exhaustion. We also noticed some differences between Chechen Tatars in Yakuts, but we cannot so far link them to particular processes. The only thing we found so far that differentially expressed genes, they have SNPs related to T2D, and some of these SNPs, they have different frequencies between different cohorts. So we are hoping that eQTL analysis or some kind of analysis linking SNPs to gene expression may help us understand the differences we observe in cell types between different cohorts. Thank you. I will be happy to take questions.
Thank you so much, Yulia. We have time to 2 questions.
Yulia, great work. Kind of curious about the some cohort study you had high levels of lipids and potentially linked to glucose levels are related to low temperature regions where high lipids may be a mode of preservation mechanism where in order to preserve from cells from freezing, in fact, you actually have a high increase of glucose in your blood, which have been shown. So I'm wondering whether that's also confounding to the fact that you have more high triglyceride levels and high propensity to type 2 diabetes in those colder regions in the population. Do you see any correlation there?
Well, we collected these samples from one place, so it's difficult to compare like a relatively high temperature and relatively low temperature. The effect of, we believe that, what literature tells us is that these people from Yakut population, when they live in their, let's say, natural habitat in the cold areas, and eat a lot of oily fish and oily meat, they actually feel better than as compared to when they come to the city and start eating sugar. It's probably linked to the way how they get obese, because these people, they don't have usually high BMI, but they have really huge waist-to-hip ratio. So I think for them, so lipids coming from the diet, for them, it's kind of protective, and their bodies are not used to tolerate high level of the glucose, actually. And at least they can do it to the level, they can do it worse than other people. Yeah.
Interesting, thank you.
I'm not sure if I answered your question, but we can talk later, yeah.
Yeah, it's a nice presentation. My question is related to your mountain regions where you said the type 2 diabetes is low. If you look into the mountain regions of people who are adopted both in Tibet and Andarian Islanders, they have a mutation, they carry a mutation in a gene called HIF2 alpha that helps them to adapt to those climate. Do you see any of those adaptive traits in these mountain people that may be responsible for limited type 2 diabetes in them?
I'm sorry, I couldn't really hear all the question. Could you please summarize?
Do they have any adaptive traits that made them
genetic traits?
Yeah, adaptive traits, inherited mutation.
Well, we checked we did some genetic work. We checked some partial polygenic risk scores linked to type 2 diabetes. And we see that despite the fact that these people are actually, they have a high BMI, and so they actually obese on average, the genetic features for the obesity are actually very, very low. So it looks like in this case, it's kind of, there is mild link between obesity and T2D somehow. So they developed this adaptation to, to split these features. And maybe this is about it, but it still requires a lot of work.
Thank you so much, Yulia. Let's applaud her.
Thank you, Julia. So I'm very excited to introduce our next speaker, Tim Stuart from Genome Institute of Singapore. Many of us know him through Stuart for the multimodal data analysis package. So looking forward to Tim's talk about scaling up chromatin analysis.
All right, thank you so much, Kwan, for the introduction. Yeah, so I'm Tim Stuart from the Genome Institute of Singapore, and today I'll tell you about some of the new methods that we've been developing for analyzing single-cell chromatin data. And I think this field is really approaching this interesting point where we can actually start to generate much larger data sets powered by some of these new experimental methods, like actually we just heard about some of this in the last session from Jiang Lan with Multi-Ancestry. And so these approaches really enable us to reduce the cost 10 or even 20-fold. And so I think over the next few years, we'll actually see the data sets becoming much larger, which is obviously really great for the field, but introduces some new computational challenges about how we can also scale the computational methods to keep up with this, which is sort of what I want to focus on today, are these two major challenges that my lab has been thinking about for how we analyze single-cell chromatin data. And the first challenge is really about the type of features that we use. So we actually still don't have a reusable feature set for a taxic analysis. So every analysis really starts by defining a set of peaks de novo that's tailored to that data set, which can work fine for an individual analysis, but actually becomes quite a headache once you start to look at public data and want to compare your data to published data sets, because everyone is using a peak set that's unique to that data set. And then the second challenge is really one of scalability, and this we tend to encounter more often in single-cell taxic analysis because our count matrix is just much larger. So for a typical single-cell taxic analysis, we might have, say, 200,000 peaks, whereas for RNA-seq, you might have only 2,000 variable genes that you're interested in. And so that count matrix is much bigger, which causes us to run into these issues of scalability much faster. So to illustrate this point about the lack of reusable features a little more, here I've downloaded 5 different PBMC single-cell taxic datasets from the 10x Genomics website. And I'm showing the peaks here for just a region of the genome. And what you can notice is not all the peaks are identified across these 5 different data sets, right? Even though these are all PBMCs, assayed with the same sort of kits by the same company, processed with the same software, but still the peaks are different. And so you can imagine how this becomes challenging if we want to, say, download all of this data and run some sort of joint analysis. Essentially, some of these data sets are missing rows in their count matrix, right? So then how do we compare those? It becomes a bit challenging. And even for peaks where they're sort of robustly identified across the 5 different data sets, if we zoom in, we can see that the precise start and end coordinates of those peaks still varies across the data sets. And so, by not having a uniform set of features, we're introducing all this additional technical variation, which is just, we don't really want that in the analysis. And so to solve this problem, actually we have to go back to the raw data and uniformly process everything. But in many cases, that's actually can be a major barrier because maybe that data is behind dB gap, or maybe it's just not available. And so this actually becomes a major problem when we want to think about how we reuse public data that's out there for single-cell taxic. So for the human genome, we can actually think about having some universal set of features where there's so many experiments that have been done already for human cells that we should sort of know what the regulatory elements are. And of course, different groups have thought about this problem, and one of them is ENCODE. And so they've put together this list of just over 1 million cis regulatory elements by looking at all the thousands of different experiments that are part of ENCODE. And there are other efforts as well. So there's this study a couple of years ago in bioRxiv where they developed this set of what they call consensus ataxic peaks, these C-peak regions, by looking at over 600 different ataxic data sets for different human tissues. And they came up with this list of 1.2 million ataxic peaks, right? And so we can just combine these. This gives us a fairly comprehensive set of around 1.5 million cis-regulatory elements for the human genome. And so this should be a fairly universal set of features that we could potentially use across any tissue, right? But the problems really start here when we actually think about how we would use these universal features in practical terms for a single cell analysis. Because here, the number of features that we have actually impacts the scalability of the computational methods a lot, right? So here, again, if we look at single-cell RNA-seq, we might have a small count matrix of 20,000 rows in that matrix, where each row is a gene, and do some dimension reduction steps on that. For a typical single-cell ATAC-seq analysis with peak calling, we would have, say, 200,000 peaks maybe, so a much larger count matrix. And then you can imagine what this looks like for the universal features. So now we have a much, much larger matrix, 1.5 million rows, and it's actually very challenging even to load this into memory and do some dimension reduction on that. So it's just, while it's universal, it's kind of too big to be practically useful for single cell analysis. But what we can notice is that many of these rows in the matrix, these individual regulatory elements, are highly redundant and highly correlated with each other. And this makes sense because we know that regulatory elements are often regulated by similar transcription factors and have coactivity across cells. So what we wanted to do is identify these sort of co-regulated groups of elements and aggregate those together to give a new set of features that we could use and reduce the total number of features that we have that way. So for the human genome, again, we can do this because we have so much data available for the human genome that will enable us to sort of group together regulatory elements based on many different experiments that have been run. So we started out by looking at over 4,000 different ChIP-seq datasets from ENCODE. So here we did a sort of broad level initial clustering of these 1.5 million cis-regulatory elements using all of this information from ENCODE. So this groups together elements that are broadly similar in their pattern of biochemical marks across all these different ENCODE experiments. But we wanted to get much finer resolution than that. So within each of these sort of broad level clusters, we compute something that we call a co-accessibility by contact score. So we compute this between each pair of regulatory elements, and this score involves a co-accessibility measurement, which we estimate using actually a very large collection of over 500 different DNA accessibility profiles that we get from many different published single-cell ATAC-seq datasets. So each profile here is actually like a pseudo-bulk cluster of cells from a single-cell ATAC-seq experiment. And then we combine this with a contact probability score that we estimate from many different high-C datasets and also the genomic distance between regulatory elements. So if we multiply these scores together, we get this co-accessibility by contact score, which is only high when the cis-regulatory elements are both co-accessible and also have some evidence for physical contact. And so using these scores, we can actually build a graph where each node is a regulatory element, and then the edge weight between nodes is this co-accessibility by contact score. And then we can just do some graph-based clustering methods, and we can aggregate, by applying this to the whole genome, we aggregate these 1.5 million elements into around 340,000 regulatory element modules that we call REMO. So we reduce the total number of features that we have quite substantially. And so, of course, we want to first test, can we use these as these REMO modules as a set of features for running a single-cell ATAC-seq analysis? And so to evaluate this, we focused on multi-ohm data, where we have paired measurements of RNA and DNA accessibility in the same cell. And what this enables us to do is annotate the cells using the gene expression information, and then we can compare if we process the DNA accessibility data using a peak matrix or using our REMO features, which one has better agreement with these RNA-derived cell labels, right? So here I'm showing an example for human immune cells, where we've processed the cells using the gene expression information, annotated the cell types. We can then do a similar analysis using the peak matrix. So here we have over 100,000 peaks for this PBMC dataset. And we can see that the similar cell types are separated using the peak matrix. So it performs fine, which is expected. There's not really an issue with using peaks in terms of separating the cell types, but these peaks are tailored to this data set. And then we can use our REMO features, and very encouragingly, we see the same sort of separation of the cell types. And here we're actually using only the top 20,000 most highly variable REMO modules. So we've actually used a much smaller set of features and get a similar separation of cell types. But of course, we can look much more quantitatively rather than just looking at the UMAP. So we focus on a couple of different metrics, both of which are dependent on these RNA-derived cell labels, so they're independent of the DNA accessibility data. So one is the silhouette, and one is this what we call this K-nearest neighbor purity score, which is how many cells in the neighborhood of a cell is the same cell type as that cell. And we can see in this case, if we process the data using the peak matrix or this REMO matrix, they perform equivalently, right? So we don't lose anything by using these REMO features, but we actually gain some scalability, which I'll show later, by using a small number of features. And these features are now sort of universal. We could apply this to any tissue. So that's obviously what we looked at next, right? So now we have the same sort of analysis, but what I just showed on PBMCs, but now for 16 different healthy tissues from the human body. And across the board, we see either the performance is basically equivalent if we used a peak matrix or REMO matrix, or in some cases, we actually see slightly better performance with the REMO features. But the real benefit here is that here we're using the same REMO features across all the tissues, whereas the peaks are tailored to each individual tissue. So with that, we've kind of crossed off this issue of reusable features for the human genome. So in the last couple of minutes, I'll tell you a little bit about the other work that we've been doing in trying to address some of these issues of scalability. So the first thing that we wanted to look at is how we actually build this count matrix in the first place. And so we actually developed a new software tool to do this that we call FragTK. So this is a Rust package that enables you to take the raw data and build your count matrix, but it does this in a very fast and memory efficient way. So here in this plot, we're looking at how the runtime and the memory usage changes as you increase the number of cells that you're quantifying for a couple of different tools. One is Signac, which my lab developed, and another is SnapAttack2. And we can plot the performance of the FragTK software, and we see that actually it's much faster, much more memory efficient. And so here for 600,000 cells, we can actually quantify that using only 3 gigabytes of RAM. So these tools are on our GitHub page, and it has the other advantage that you can just download a single binary, and that's it, you're done for the installation. We also wanted to enable a sort of on-disk workflow for analyzing very large data sets. So we developed another Rust package called Sparse. And what this enables us to do is actually compute these analytic Pearson residual variance scores for each feature and do feature selection. So this feature selection step can happen on disk. So we can subset the matrix on disk using this Sparse package. And then, for very large data sets, we can actually subset the matrix on disk and load in a much smaller data set and do downstream processing on that. So to illustrate that, we looked at now a much, much larger collection of cells by combining a couple of Atlas data sets for the adult and fetal human. And this gave us 1.2 million cells to play with. And we can look at how the runtime and memory usage for doing dimension reduction step on that data set changes if you use a peak matrix or you use our REMO features. And you can see that there's a pretty substantial difference both in runtime and memory. And you'll also notice that the line for the peak matrix actually ends at 400,000 cells, which is because the peak matrix actually gets too big to be loaded into memory and constructed with more than 400,000 cells when you're quantifying a peak matrix. Whereas for the REMO features, we can actually scale to the entire 1.2 million cells, which is what I showed on the previous slide, and get that done. So some nice gains in the performance when using the Remo features because we have a much smaller number of features. Okay, so with that, I think I'm out of time. So I just want to thank all the members of my lab, especially Chrisania Lim, who has been driving this Remo project, and our funding and support from NRF and A*STAR. So thank you.
Thank you very much, Tim. Do you have a quick question for Tim?
Hi, Tim. Really great tools. I was just wondering a technical question. So regarding the sparse matrix, so in R, there's kind of up limit of like number of non-zero and zero number in the sparse matrix. Can those two like surpass those kind of limits and scale up to, let's say, even 5 million or 10 million attack-seq data?
Yeah, so the limitation on the size of the sparse matrix in R, so we don't tackle that, but we just kind of get around that by making a smaller matrix. But there are other matrix classes that you can use in R. Those tools are just a little less developed than the main like sparse matrix class, but they do exist. So yes, you actually could construct this much larger peak matrix and do something with it, but it would require a lot of RAM. I think for scalability to like tens of millions of cells or whatever, we're a bit more interested in the more disk-based matrix objects where you load data in small batches into memory. But if you have the RAM, like you could think about like 64-bit sparse matrix classes as well. Yeah.
Cool. Thanks.
Thank you, Tim.
Sorry, we don't have time.
Okay, thanks.
I'm so sorry. So for the next talk, I would like to invite Varodom, Charoensawan, and he's going to talk about systemic immune profiles in Southwest Asia population.
All right, thanks so much for very kind introduction. Since I think this is toward the end of the day, and so I just want to keep it very, very simple. So there are only two things that I want to cover in this talk, and I'm going to try to make it very simple. So the first thing, I guess I'm the only speaker in these sessions from Thailand, so I feel compliant to maybe talk about our little but maybe significant contributions to HCA, as a country. Sorry, Thailand as a country, but to HCA as a whole. But also the second point is just to try to follow the theme of this global science and local stories. So I just want to show you just one UMap, that's a promise, of how we try to use the data that we generated with the HCA and try to ask the question which is relevant to Thailand, but also hopefully it's related to global implication as well. Okay, so I'm not trying to follow what Kwon was doing, but yeah, I mean, I think we were lucky that we got the fundings together with Sarah. Actually, if she still remember the Newton Fund. So we used that as a leverage to basically get the first access to some of these technologies, and basically as a developing countries, I don't know if others experience the same thing. It's quite difficult to get this so-called expensive experiment. And also tools, and that was possible because the generous offers and support from many grants and also people. So I just want to give a special thanks to Sarah, which is here. I think this is the first time that I'm giving a talk in front of you after this 15 years. So by the way, yeah, Sarah's my PhD student 15 years ago. I don't think I've met her in 10 years, so I'm a bit sweating right now. But anyway, everything started, with the collaboration from Sarah, and I think this is about time when we visit, and most of the postdoc and people here, they become a PI already. But yeah, anyway, they're not forgotten all the help from that day. So since then, so all those tools and equipment that we have is not there just to show off. So we try our best to make it accessible to as many people as possible. And in Thailand, we try to make it keep it open. And since then, we have lots of collaborators, and we have application in different fields. Well, Pornpan, who's already here, and you probably heard her talk about like how we use single cell to study local but significant problem like dengue infection. And yeah, I'm sure you probably heard her talk, and she would tell you more in the future. And then our small contribution to HCAs, this is dated back to before COVID, as many of you might recall. I had the pleasure of attending this HCA conference in Addis Ababa just before COVID, and that's how I got to know Sham, Jay, and how and Wun Yang as well. So that's how we got into IDA. And then, but I think we as a small country, we're doing pretty great in terms of contributing to SCAs. We also hosted SCA meetings just after the COVID, so I think that was quite was pretty concerning if Pan still remember, because we didn't know that it's going to be turn into online or not. Is it's going to be another outbreak or not. So that was pretty fun. And then just last year we also hosted another SCA workshop. So that's a few things that we think we committed and try to contribute to SCA as a whole. So maybe okay, so I think I'm doing pretty good with time. So I guess I have some time to tell you a little bit story size-wise in terms of how we got into these communities, and also contribute something as well as ask a question that we're interested in. So together with Sham, Partha, Jay, and Wong Yang, we managed to land this CCI fund for immune diversity at last. So you heard about this a few times already this day, so I'm not going to go into details. But what I want to share with some of you who may be from so-called developing countries as well, is that it's really difficult to find like fundings to run this kind of experiment, right? Living alone, very like population case. So one thing that we managed to do, and this might be useful for the other, is that once we got this funding from CCI, we used that as a leverage to talk to our nationals funders in PMUC, and basically they match the same support. And I think that's how we basically able to generate and put basically Thai populations on the map, and with IDA and so on and so forth. Okay, so right, so I think I just want to introduce sort of I would say like local niche, but then I think it might be interesting of some of you. So Thailand, we situated. So I guess like we all know we're here, right, in Singapore. So Thailand is a little bit further up north, and you can see from this map that we right in the middle of mainland Southeast Asia. So there's quite a lot of a bit of story that you might be interested here. So we call this region Indochina, and I guess you can guess for obvious reasons. So we have India here and we have China there. So we're right in the middle. But also, I mean, I try not to touch the politics thing a little bit, but well, in the past, during the colonizations, so on the west, right, Myanmar and Malaysia were colonized by, British Empire. I think that's the way to say it. And on the right-hand side, to the east, right, that's all the French colonies. So Thailand was kind of like buffer state in the middle. So basically, that's quite a bit of story, how people travel and not travel during that time. So one of the things that if you ask any Thai at all, right, where you're from, you would have lots of different answers. And actually, this is something that there's no consensus whatsoever, right? So I think Shyam and Kin Hong, you might remember, you asking us like, can you identify like the ethnics, like something similar to like in Singapore, right? You have Chinese, you have Indian Chinese, right? Sorry, you have Indian and you have Malay. You don't have that in Thailand because everything sort of mixed up, right? So the theory is that there are quite a few theories. There's some story that people in this region might be from the north of China. Maybe they're already here somehow, and then maybe South China. But anyway, so if we look into the sort of like the current hypothesis is that usually, I mean, they think that there might already be some people in this region before, showing in blue here. And then there's some migrations from the north. So I like to call it from the north because previously I used the word Chinese, but that might not be correct. So but anyway, you can see that as from the ancient DNA, you see like more and more proportions of the green proportion components coming downward. Also, we see lots of different waves of genetic flow between the north and Southeast Asia. But also, this is something that has been discussed and argued for a long time. And well, I mean, quite a few years ago, 2018, the two papers inside back-to-back just talk about how complex and how complicated, diverse in terms of genetics of these regions. But also, just less than 2 months ago, there's another paper in Nature on this particular topics. But obviously, this is genetics. And so, well, as we being part of IDA, or we call IDA 1.0, this is the papers that Xiao and King Hong published, I think at the beginning of this year. So this is a subset of Thai that we collect. So back then, we start collecting sample for the first time around 2020. And then, so you can see that, we have group 1 that we collect just before COVID outbreak, and also we have some COVID-19 vaccinations, and then we finish everything in group B, batch 3 and 4 after the COVID. So in a way, yeah, I mean, it's not like we intend to do that, but that's something we can look into as well, like the difference between before and after. But same as other IDA samples, we done genotyping using SNP array and 5' GEX, and also we done TCR and BCR as well. Okay, so I like to call this like let's take a first look into the immune profiles of the Thai and people in these regions. But so the first thing we've done, of course, is just to done like PCA of look at the genetics. So it looks as we expect that Thai being in the middle, showing in pink here. So we can see that Thai is also situated in the middle between the Indian and East Asian populations. The Malay probably a little bit closer to India, but yeah, we kind of like right in the middle together. So that's in terms of genetics. But also, if we look into the admixture, you can see, I believe, I see this interesting trend that all the Indian, even though, from India and also in Singapore, they look pretty similar together, I mean, in orange. So I chose the color myself. So if this is not satisfying, I mean, I take all the responsibility, by the way. So you can see that, Malay, Thai, and Chinese, we have a mixture between the red and the blue ancestry, which I would tell you what we think about that. So you can see Japanese mostly green, and Korean mixture between red and green. So kind of middle between China and Japanese. So I think I see nothing from the under, right? So you agree that this is the case. But also, I just wanted to say that that's not just something that we see in the IDA. So we mix that with 1,000 Genome Project. And this is to confirm that what we see in India, right, is the same as India from 1,000, same as Chinese and Japanese and so on. You can see Thai right here that we have a mixture, and in terms of like mixing between the red and the blue, it's really well mixed. So you see the range of red and blue. And in fact, if you calculate like the admixture index, is highest in Thailand. So that's just to confirm that this is a real melting pot that everyone just marry everyone. So as I said, there will be only one UMAP in this talk, right? So yeah, this is from Yulia's talk and other people's. This is the same talk, so same plot. Something that we keep saying, and I didn't know that Yulia would talk about monocyte, but that's something we observe as well. So for example, right top there, you see that in terms of from single cell data array, we see some discrepancy in terms of the proportion of monocyte. And you might think, okay, that might be, just maybe different countries, different, batch effect and so on. But you would see that, in CBC, when you do the blood test, we see exactly the same thing. So there's something there, right? And that's from Ken Hong's papers in Cell. So what we're trying to see, right, is that we talk about there's a component of, mixture between at least two components between the so-called red, probably migrating from the north, right, or the blue, which probably already, be in that region before. And we can see that, so we look into, done differential abundance analysis. So the three countries that we're showing here, right, are the ones that have a mixture between blue and red. We can see that only in Thailand, we can see certain cell stage in CD14 monocytes. So that's, we talked about that a few times. As the Sure. Because, I mean, basically, we have a so-called self-report, right, different countries. But if we look into like the proportion of, the rate ancestry, we can see more, enrichment at certain cell stages. So why is that the case? I'm not going to try to make any conclusion here, but there could be lots of reasons. So obviously, there's a link between, like the enrichment of rate ancestry and, higher cell stage in for the CD4 monocytes, and would that, can we subpopulation? Can we tell exactly what's the cell stage? Also, because this is, RNA-seq, so we can do gene expression to see what exactly would be the markers. But also, I'm not going to try to, over interpret. But yesterday we also heard a great talk from Yusuf about the link between malaria infections, right? And I think CD classical and non-classical monocytes. So we think in Thailand we have a lot of people who have thalassemic traits. So might not be the active infections, but could be something that related to the blue ancestry who's already in the regions before the red comes in. But also there could be other things that we can look into like EQTL and so on. So next step we're going to add like more samples into this, and the next time that we do is in IDA 2.0. So we're recruiting people from different regions, and we're checking their birthplace, current address, and so on, to make sure that they, not influenced by moving too much. And you can see that people from different places in Thailand, they dress differently, have different cultures, they have dialects, and we believe that to a certain extent they represent countries, neighboring countries around us as well. So in a way, it probably would not just be Thailand, but we can, take into account, people around Thailand as well. All right, so just want to quickly, acknowledge, everyone who worked on this. So Serentip, Lamita, Narita, they mainly help a lot, with the data generations. So Benjamin Pon is my PhD student who worked mainly on this. The next time you're going to hear about this story, it will be her to present this, and also with help from Serentip and Prawa on this. So and also I'd like to thank all the IDA families, not just Jay, Wong Yang, Sham, and Partha, but everyone, Ken Hong, and we have I think bi-weekly meetings, which is even more frequent than some of my meetings, and also our IDA teams. And if I could just spend like 30 seconds, give a shout to my wife, because with the two little monkeys, I haven't been traveling a lot at all, and I asked her, this is the one that I really want to come. So yeah. And the day that I left Thailand, the little one got COVID, so it's extra work for her. But you can see that it's kind of like mixing between the red and the blue here as well. So it's like you do experiment every day in your life as well. So yeah, and thanks for giving all the time. Thank you.
Thank you, Varodom. It was a really great talk. Unfortunately, for interest of time, we have to move on. Sorry.
Thank you, and I'm excited to introduce our next speaker, Dr. Yasuhiro Murakawa from Japan, and he will share about the 5' single RNA sequencing protocols and data sharing. Over to you.
Thank you for the kind introduction. Yeah, I'm Yasuhiro from Kyoto University and RIKEN. Today I'd like to talk about the 5' single RNA-seq methods and data to empower the community. So from my side, this is the overview of what I'm talking about today. So I like to emphasize that although 3' single RNA-seq data is often used, 5' single RNA-seq data can also tell us a lot about not only about gene expression but also cis-regulatory elements, splicing events, and immune repertoires. And I also like to talk about our own examples in which we generated an atlas of transcribed enhancers across helper tissue diversity for decoding human immune-mediated diseases. And also I'd like to emphasize that a large-scale 5' single RNA-seq data have been generated in Asian countries. Speaking about enhancers, one of the key elements to regulate gene expression. So upon activation of enhancers and promoters, transcripts happens from genes. And 5' end of transcripts are capped and introns are removed by splicing, and polyadenylation happens at a 3' end. So for this, we are mapping transcription start sites of RNAs to study gene expression and also enhancers. So the reason for this is because we all know that mRNAs and non-coding RNAs are generated from genes, but also very interestingly, active enhancers produce bidirectional enhancer RNAs. And by studying the transcription start sites, we can identify enhancer regions at high nucleotide resolution. So we have been working a lot to identify a lot of groups from Phantom, including Jay also studied a lot to study the TSS, transcription start sites. And to do this, one method to do this is to look at the cap structure added at the very beginning of transcripts. So this is 7-methylguanosine cap added at the very beginning of the transcript. And by specifically capturing this the cap structure, we can analyze TSS. And one such method to do this is template switching. The way how template switching works is we use oligo-DT oligo and also template switching oligo. And then if you do RT reactions, the cDNA synthesis go over to the 5', and then this template switch reaction happens when more preferentially when cap is present. And then add additional preferred sequence. And then we can sequence the 5' end. So this is one such method to do this. And actually this template switching method is used in 10x kit. So the way how it works if you use 3' kit is you add oligo-DT conjugated to UMI and cell barcode to the beads, and then template switch oligo is added, and then if you do RT reactions, and then cDNA goes all over to there, and then the fragment the cDNA, and then the final cDNA, NGS library is generated like this. But if you use 5' kit, it's basically the same thing. It's just the difference is beads added to the template switch oligo. But we use also oligo-DT, and then RT reaction happens. So basically the same method, but then NGS library is generated close to the 5'. And then NGS, if you do sequencing, sequencing actually read 1 is used to only sequence UMI and cell barcode, but read 2 is used to count and sequence transcripts. So if you use 5' kit, actually if you just use read 2, it's only sequencing the vicinity of 5', not directly the 5' end. So read 1 is only 26 base pair long. So what we did is in the 5' single cell RNA-seq, so it's very important to do a long read 1. So not just sequencing the cell barcode and UMI, but also go beyond the template switch oligo and reach the exact position of TSS. And we actually did this, and then what we notice is the reads that arise from the true transcription start site have additional G at the beginning of the transcript that are not encoded in the genome. And what is this? And this is actually coming from the cap. So the cap seems to be reverse transcribed to C, and then if you make cDNA, and it has additional G. So we use this signature, this additional G, to distinguish whether this is from the true TSS or not, because RT reactions can also stop when, for example, you have a GC-rich regions. So to only get the right TSS, only the reads that have this unencoded G can be retained, and then the fully matched reads can be thrown away. So by doing this, for example, this is looking at the all reads compared to unencoded G reads, and if you use unencoded G reads, you can filter out a lot of noise, but still there are some regions that have this bidirectional transcription in the intergenic regions. So if you zoom in these regions oftentimes overlap very well with active open chromatin regions. So just simultaneous analysis of gene expression and active enhancers can be done only from RNA analysis alone. So this filtering procedure and calling bidirectional transcript enhancers can be done using a RipTech package. And then we did this analysis to 1 million human helper T cells, to 1 million helper T cells. So this analysis, these T cells were collected from healthy donor, actually only 3 people, but 100 ml of peripheral blood was collected several times and FACS-sorted, and then we FACS-sorted Tregs and TFH and subpopulations. And then we generated this multimodal data sets comprising of not only 5' single-strand-seq data, but also the mass-seq and single nuclear ATAC-seq and also 5' site-seq to get not only a transcription start site or gene expression, but also the chromatin accessibility and surface protein expression. And all this data set is actually uploaded to the Human Cell Atlas data portal 2 days ago. So thank you very much, Ida, for very kind help. Yeah, so this is how the data briefly looks like. So the data contains not only for the bulk CD4 positive T cells, but also FACS-sorted subpopulation of T cells. And from all these 100 million helper T cells of 5' single-strand-seq data, we identified active enhancer regions in each T cell subset. And we identified more than 60,000 active enhancers, and we integrated with large-scale GWAS datasets to identify enhancer regions that directly overlap with GWAS SNPs. Ended up in identifying 606 disease enhancers across a variety of autoimmune and allergic diseases. This is one example of IBD enhancer that contains SNP for IBD. And if you activate this enhancer using CRISPR activation, which is done by Yoshitomi Sensei in Ueno Sensei's lab, it's very interesting that you can clearly see the induction of enhancer RNAs followed by also the clear specific induction of mRNA from the IL-7 receptor genes. So this connection of enhancer and promoter were predicted using micro-C chromatin contact maps. And even if you look at, even when looking at globally, IL-7 receptor gene is specifically upregulated. Yeah, so this is a brief overview of our studies. So we can use this 5' single-strand-seq data not just to count gene expression, but also to look at active enhancer regions, and then to specifically narrow down the disease enhancers and to look at the target genes of these disease enhancers to get a hint at the genetic mechanism of susceptibility. So this is one of the take-home message. The 5' single-strand-seq data is very powerful. That it also tells us about the accurate transcription start site, and this leads us to not only just gene expression, but also bidirectional active enhancers. Also by simultaneously analyzing and correlating mRNA and eRNA, we can also study enhancer gene regulatory network, and also you can do for immune cells, immune repertoire analysis. And then looking at this AIDA dataset led by Jay and Unyan and Sham. So this dataset also contains a large-scale 5' single-strand-seq data, which is very nice, and also very beautiful publication from Boshan's lab last year. The 5' single-strand-seq data was used to look at the splicing events because the last exon, which is close to the 3' end, so it's a huge exon that mostly contains 3' UTR, not the coding sequence, but 5', it has more splicing. So the 5' single-strand-seq data can be used to also infer splicing events. Yeah, and also Jay and Chuns are also leading this 5' single-strand-seq data from various tissues. So this is a very fantastic ongoing effort to map cis-regulatory elements in the human genome using 5' single-strand-seq data. I also like to thank all my collaborators. Without them, this project have not been possible. Also, I think to think about the community efforts, I think original technology is also very important. And here, the 5' single-strand-seq data have been developed and then in large collaboration, like Jay and a lot of groups, and it has been really analyzed very nicely, I believe. So with this, I'd like to thank all of you and for this chance, for all of you, and thank you once again.
Thank you. We have time for a few questions.
Thank you. Thank you for the great talk. Since you are targeting the 5', you managed to capture all the long non-coding RNAs because they can be polyadenylated or not.
Yeah, exactly.
Did you explore the long non-coding RNAs in a single cell perspective?
Yeah, the great point. Yeah, we could actually get a lot of link RNA as well. And one of the reasons also is because the oligo-DT primer, it can also internally hybridize internal A-rich regions. That seems to also capture not only the polyadenylated transcripts, but also non-polyadenylated transcripts, including eRNA. So, but link RNA, we could get a lot. And we also performed mass-seq to get the long-read single-cell RNA-seq. So that also identified a lot of novel link RNAs expressed specifically in T cells. Thank you.
Interesting talk. My question is, can you use this to characterize bivalent genes in a cell type specific manner? Do they function in a bivalent genes?
Bivariant in terms of activation status?
Yes.
Yeah, I think that's a great point, actually. We also looked at the ATAC-seq data. So the chromatin open regions is also analyzed, but still, transcription is not really happening.
Yeah.
So there are many cases like that. So I think I think the combination of single-cell RNA-seq and also ATAC-seq, for example, that will help us to look at bivariant transcripts as well.
Thank you. I may have a question about when you use this protocol to look at the transcript isoforms, for example, like the exon structure. Like, would you be able to tell like the structure of a long non-coding RNA that is relatively unknown, and how confident is this to study the novel transcripts?
Yeah, thank you. This analysis actually may be more preferable to, not a long link RNA, but we could actually use this data sets to infer the structure of long non-coding RNA as well. Looking at the cDNA length distribution, it was about less than 1 kb. So it's not the full length with the single cell resolution. It was more or less like a long read, longer than short read. So we also use a bulk long read as well. So if you do bulk sequencing, you can optimize cDNA construction procedure so that you can get like a much longer, like a couple of kilobase transcript can be annotated in long read. So maybe the combination of long full length analysis using bulk analysis, but also combining with a single cell to look at the expression specificity. So I think this kind of like combinatorial analysis of bulk and single cell, that would help us to get a deeper insight into the structure of link RNAs of long ones.
Yeah. Thanks, Dr. Yasuhiro again.
Yeah, thank you very much.
Thank you. So it is my pleasure to announce the next speaker, my friend, Dr. Vinicius Maracaja-Coutinho from University of Chile. And he's going to talk a little bit about the Chilean results associated with the Latin cells, right?
Thank you. Thank you very much, Mari. Good afternoon, everyone. Today I will talk a little bit about the single-cell research environment in Chile and how we managed to build a community there for single-cell research. Chile is a South American country with over 4,200 kilometers from the north, Atacama Desert, to the Patagonia in the south. In Chile, we have different extreme environmental variation, with people living from the sea level to high altitude ends, and remote territories like the Atacama Desert or Rapa Nui Island. It's a country with about 20 million people, including a wide, diverse set of Native Americans and mixed populations, resulting in a rich setting to explore how environmental and ancestral diversity shape biology. However, the situation for single-cell research a few years ago in 2019 in Chile was similar to other regions in Latin America, with minimal representation or almost no representation in the human cell atlas community. So the local community was completely absent from the global discussions related to single-cell research and the human cell atlas. And also we had the local scientists, they were completely unfamiliar with the concept and the potential of single-cell genomics. And we also lacked both infrastructure and human capacity in the country. So we had to do a very strong capacity building locally in order to start to build a community, even teaching what does single-cell biology means. We started with some spreading the word on single-cell research through courses, symposiums, and seminars. For example, this is the first symposium meeting dedicated to Latin America. In that opportunity, only 7 speakers based on Latin America participated. And in that opportunity, we organized working groups after some breakout sessions with community from all the continent in order to discuss how we could improve the participation of Latin America in the consortia. And in Chile, over the years, all the team together, we managed to organize different courses in the upper and symposiums over the years in the continent, and some of them were organized in Chile. For example, the first data analysis workshop organized by the Human Cell Atlas was teams from Mexico, Chile, and Brazil. It was an workshop devoted to this population, to the Latin American population, scientific community. And then years later, we organized the first wet lab, hands-on wet lab data analysis in-person workshop in the continent. It was in Santiago, Chile, a few years ago in 2023. We then organized several other data analysis courses in local languages, and the next one we'll have in July next month, organized together with the regional student group from the International Society for Computational Biology and the Chilean Bioinformatics Society. We also organized several symposiums. For example, in last year, we had a hybrid human cell atlas symposium dedicated to Latin America. It happened simultaneously in Brazil, Chile, and Mexico. And we also organized, combined all these activities with seminars in local language for the local community in the country. And we also, another very interesting way to reach the community locally was to organize mini symposiums inside the national meetings of local scientific societies. For example, next October, we'll have a mini symposium dedicated to explore the cellular diversity and all the advancements that we have in single-cell genomics from Chile. That was a very interesting way to reach people locally to get involved with the Human Cell Atlas. And over the years, we were developing a lot of open training material covering different aspects from single-cell data analysis, since the very basic analysis to more advanced analysis, like alternative polyadenylation in a single-cell perspective, spatial transcriptomics, etc. We developed a total of 12 Google Colab notebooks. We also developed some Jupyter and Docker versions of this material. All these material were made available in 3 languages, English, Portuguese, and Spanish. They are being used in different courses organized by the Human Cell Atlas and also by the Wellcome Connecting Science from the Sanger Institute, and discussions are occurring in order to make this material available in the Human Cell Atlas web portal. Several courses over the world is already using and reusing this material, changing, modifying, and replicating. And this material is completely open. You can use, reuse, and do whatever you want to train people in single-cell data analysis. In Chile, we have a very well-established funding ecosystem. And with this transition of the Human Cell Atlas from the health atlases to disease atlas, I believe the country will be able to contribute even more to the consortia. For example, here we have some different levels of funding. The funded seed grants, that they are personal research funding from 3,000 to 6,000 euros per year for 3 or 4 years. We have in the middle the network research funding that are focusing on building a community, building a network of researchers in a particular topic of interest with the potential goal in the future to construct a virtual center, advanced center, about 800,000 euros per year for 3 years. And we have in the other part of the screen, the National Advanced Center for long-term funding, about 1.5 million per year for 10 years. And also we have some funding dedicated specifically to scientific, to medium size or large size equipments. For example, this year we applied for a Xenium platform through this kind of funding. And here we can see some of the examples of the national advanced centers that were formed in the country with Geroscience. Gero is dedicated to study aging. We have the Instituto Millennium of Immunology and Immunotherapy dedicated to immunology. The CCAN, dedicated to cancer, the Center for Genome Regulation. We have the Advanced Center for Chronic Disease, focusing on gastric cancer and cardiovascular disease. I am one of the PIs of that center, among others that we have in the country. And the country is also, because of these centers, we have many established biobanks over the country, with some of them part of the Latin America biobank network. For example, in the University of Chile, we have the Banco de Tejidos and Fluidos. That's one that we are using a lot in our projects. With the grants that we received from the Ancestry Network for the Human Cell Atlas from the Chan Zuckerberg Initiative, we managed to establish a new advanced genomics unit in the University of Chile with all the cutting-edge equipment for sequencing and generating single-cell research. We also in the country have other facilities from universities and also some companies with officers there. So we have all these infrastructure available in the country at the moment. And also talking about the computational infrastructure, in the country we have the National Laboratory for High Performance Computing. Is completely open for Chileans. You just need to submit a project and you can have full access for storage and data processing for free. And in Chile, since we're talking about this transition, the Human Cell Atlas for disease, these are the most frequent causes of death in Chile annually. Some really interesting opportunities for single-cell research. The top disease we have cardiovascular disease, cancers and respiratory disease and digestive disease. I'd like to talk now about some of the interesting local flagship projects that we have at the moment running in the country. The first one, for us, of course, me, Patricia, the gold project for us, is the Latin cells, a human cell map of Latin America diversity. That's the CZI grant we have through the Ancestry Network for the Human Cell Atlas. In this project, the first goal is to generate a human cell map of indigenous and admixed populations from different countries in the region, from Brazil, Chile, Colombia, Ecuador, Mexico, Peru, Uruguay, and Caribbeans in the US. From Chile specifically, we already sampled 80 individuals, PBMCs from 80 individuals from 4 different ancestries. Here you can see the genomic variation from some of the samples that we have. And here in this part of the screen, you can see the Chileans. And we have a very interesting case here of the Aymara population that we can see half of them with a very complete indigenous background and other with an admixed background. This is a very interesting population that we'd like to explore a little bit more to see how they evolve, for example, for exposition of disease or even to vaccination, for example. So far, these are some preliminary data. We managed to integrate approximately 1 million cells from 235 individuals so far from these 4 countries. And here some of the comparisons that we made recently, comparing, for example, Chilean Aymara ancestry against the Pastos ancestry from Colombia. Here, each color is a particular cell type, each point is a gene, the logged fold change, and the size of the dot is the percentage of cells expressing that gene. We did this kind of analysis for all the ancestry from all the countries that we have. Here are just some examples, a comparison between the Chilean Aymara against the Maya from Mexico. And the second goal of this project is to create a gallbladder tissue cell atlas. In Chile, this is of particular interest. There we have the Chile map, the map of Chile in my right side. You can see in red scale the Mapuche ancestry over the country. We can see a place there that's more dark. This is the Araucanía region with the higher incidence that you can see there of gallbladder cancer and also the high incidence of Mapuche ancestry. In the north, we can see the less incidence of gallbladder cancer and also the less incidence of Mapuche ancestry. It's the region with higher incidence of Aymara population. And these, there is a hypothesis that this is something, the gallbladder cancer is something associated to this Mapuche ancestry in the region. And this is a rare cancer, but in Chile we have one of the highest incidence globally. And for gallbladder cancer, we already sampled 64 individuals from admixed Aymara and Mapuche ancestry. We already we just finished the optimization of the cell disassociation protocols, and the first samples are being sequenced right now. Some other flagship projects through the network funding that I mentioned before. We managed to approve this project, the Inflammates, that we are building. We already built actually a cohort of people living with HIV in the country. We are doing some multi-omics approach. This is we want to compare people living with HIV with and without metabolic syndrome. We compare it to people without HIV with and without metabolic syndrome. And we have access also to children for the next phase of the project that we are at the moment fundraising. Other projects that we are also part of at the Advanced Center for Chronic Disease, we are following up a cohort of 10,000 people since 2014 from the region of Maule. This is called the Maule cohort or Malco. This is the region with the high incidence of gastric cancer in the country and the high incidence of cardiovascular disease. We are planning now to run some single-cell assays in heart failure individuals and gastric cancer. This cohort started with healthy individuals that over the years they started to get sick. Other projects that we have, other flagship projects we have in the country, this is led by Dr. Mercedes Lopes. She established a cohort of long COVID and a cohort on a biobank of long COVID. She published the first paper using only flow cytometry into 2021. At the moment, she is about to start some single-cell assays. Another very interesting project by Dr. Cecilia Vial and Dr. Juan Calderan is exploring the infections of antivirus. This is of particular concern in the south of Chile and south of Argentina. And they are about to start the single-cell assays. With Dr. Alejandro Corvalan, he established a gastric cancer cohort of thousands of individuals in the south of Chile with several different biospecimens available, and they are planning to start single-cell assays very soon. And in the computational field, we also have some interesting projects. For example, this is one of the projects that we have in the country, led by Dr. Alberto Martin. He developed this tool, HCA Explorer, is a user-friendly platform to perform the entire step-by-step analysis, single-cell data analysis. And this research is already impacting, this research impact is already being reflected in the publications, as we can see here, an increase in the publications when we search for single-cell publications from Chile in PubMed. Thank you very much.
Thank you, Vinicius. We have time for one quick question. Yes.
Thank you. It's a question about what can we do from UNESCO to you. We have the science offices in Latin America, and we organize events like the CILAC, the Forum for Science. Do you think you can use this partnership with to catalyze your network? Thank you.
Yes, definitely. I believe the branding, as we discussed it in the lunch, the branding from UNESCO would be of key importance to raise awareness to this initiative and to try to put together efforts from the different countries. I'm talking not only about Chile, but Latin America and the Caribbean in general, right? Something that I was discussing with John is, for example, trying to approach funding agencies like the Gates Foundation. In the past, they were a little bit hard to have access, but I believe that the reason was because the Human Cell Atlas was targeting healthy individuals. And Gates Foundation is more focused on neglected disease. And in our continent, we are full of neglected disease that would be an interesting target. As I mentioned yesterday also, we managed to receive a small funding to organize a United Nations University course in Costa Rica, maybe together with UNESCO, try to fundraise this kind of, obtain funding for this kind of event, not only course, but symposium over the country, because it's always complicated because we don't have access to funding, but of course, to launch a course, we don't need too much money. As Ricardo is here, we organized some courses in Salvador, in Brazil, without money, We only managed to have funding to move some people in person there. Others were live, giving some introductory classes, but their hands-on training were led by the people locally. We were how many people? 3 people working there, and all the other speakers were online. So, of course, this is not the best situation because we are with too many work to do, a few people, but with a small amount of money we can do. And I believe that UNESCO could work together with us. I understand that UNESCO is not a funding agency, but can catalyze, as you mentioned, how to fundraise to organize this kind of event. And also, just a last comment, maybe work together trying to organize we could work together to organize some science policy events, inviting the decision makers to be together from different countries. In Chile, Chile is part of EMBO, the European Molecular Biology Organization. And they mentioned that they are interested in funding this kind of event. Maybe the Human Cell Atlas, UNESCO, together with some institutions in the continent could apply for, I don't know, a 3 days event, just discussing something like that. I have some ideas.
Thank you, Vinicius.
Thank you. And the final talk, we're going to learn more about single-cell projects happening in Brazil. Welcome, Dr. Patricia Severino.
Hi, everyone. So for this last session, I'm going to complement a little bit what Vinicius said on some directions in Latin America. I'm going to be showcasing Brazil, but it represents a lot of what goes on in Latin America, okay? So it's about landscape challenges and future directions. So single-cell research in Brazil, there are big opportunities for research over there. So we have been talking about this global gap in single-cell RNA-seq diversity. So most single-cell atlases focus on Northern American populations, or Northern Hemisphere, to be more precise, and Latin America is underrepresented. So the disease burden in Brazil, there is a high incidence of tropical and neglected diseases like dengue, Chagas, etc., and they're driven by endemic conditions. The reason why I'm bringing this is because I think we have a lot of overlapping with other Latin American countries, but also Asia. So I think this should set some of the things we should be discussing with Asia for collaborations. So biomedical potential, single-cell RNA-seq and decode host pathogen interactions and immune responses, which are very important for us locally for applications, especially in vaccine development. Here I'm giving you some examples of what is this disease landscape and research gaps concerning neglected tropical diseases. So we have high incidence, some of them, a lot of very high burden of others. There are multiple studies, but what's being done with single-cell RNA sequencing is still very little. There are some diseases like dengue and tuberculosis where we get some funding, but not for single-cell RNA sequencing locally yet, and others like really severely underfunded like Chagas, schistosomiasis, and malaria, despite the heavy, very, very high burden that we have in Latin America, also Asia and Africa. If there is anybody here from the African network that we can also discuss this. So the distribution of research on neglected tropical diseases locally. So this is the funding scenario in Brazil. Brazil invests less than other countries that have similar burdens. So, and of course of more high income countries. So we believe that these funding gaps offer an opportunity for joint projects because there is a lot that can be done together, so we can gather funding together. What else? Here I wanted to show you what Brazil looks like. So the map here shows that we're divided in regions. These regions are like divided like that due to social economics and also kinds of health issues. So that's how Brazil is divided. Our public research funding is very, very well structured. So we have a lot of national opportunities, but also opportunities which are divided among the states here. They're called foundations, local foundations. And especially in the southeast of the country, there is a lot of opportunities for funding. funding. Does not happen in the other regions of the country similarly, but we can always partner with them to tackle diseases that are more significant in these other regions. So there are quite a few. What I wanted to show here is that we have different layers of funding in Brazil and opportunities. So we have the national public agencies, we have the state level support, especially in São Paulo, where we have a lot of genomics and also biotechnology going on. Then we have public health institutions, where there is a lot going on in terms of infectious diseases research, and external resources. So there are a lot of funding that come to Brazil, and we can partner with, and the idea is when we put together a project, we have to take a look at all these opportunities and engage with them together. And there, when we talk about specific institutes that have infectious diseases research as opportunities, they are very much engaged with all the local communities and all the local possibilities for sampling. So the idea is that this cannot be done under one layer. You have to talk about the ideas with all of them so that we can have enough funding to do it. So the strategic financing models would be exactly in this way, to take a look at all the possible layers to be able to finance projects with the amounts that we need for single-cell RNA sequencing and other single-cell biologies technologies, right? So next to the neglected diseases, I wanted to bring here one example of other things which are like more easily funded in Brazil, which are diseases related to the top causes of death. So the Brazilian population is aging. So this is what I wanted to show you here on this map. We're currently here in the middle, but this means that we're aging fast. And on the right side, top causes of death for female and male. You can see that it's mostly heart-related diseases, and similarly to what Vinicius showed in Mexico, diabetes and so metabolic diseases, stroke, so everything related to cardiovascular and hypertension. And so I'm showcasing only 2 projects, not the same as Vinicius showing all the things that they're doing in Chile, but I'm just showing 2 that are based in São Paulo, and there are very big opportunities for collaboration. So this is a cancer for aging research. It's coordinated by Marcelo Mori from the University of Campinas in São Paulo. Mariana, who is here, is also part of the group. This is how we have organized ourselves. So the mission is to understand how aging enables disease and finding ways of prolonging like healthy lifespan, or revert aging, which is like what I suppose many of us would be interested in. And aims, so general aims, identify common mechanisms leading to age-related conditions, identify early-stage dynamic biomarkers that can predict biological age progression and possibly revert that, find ways to prevent and treat age-related conditions as a whole, and identify environmental and lifestyle factors influencing disease. So this is a project that has just started. This is how we have organized ourselves. Each one of these little circles is one researcher, and how we collaborate with each other in different areas related to aging. So I believe that everything that everybody does here can be inserted here somewhere, you know, and everything that everybody does is in somehow related to aging. So I think it's one way of looking at everything that we do and maybe finding ways to collaborate. When we're talking about diversity and different ancestries, we have a very diverse population, like I showed you. All of these people come to São Paulo. Our cohort is in São Paulo, so we have a lot of possibility of studying environmental and ancestry background, but everybody who is here also, because Brazil has ancestries from everyone who is here, 100% sure. I don't think there is any nationality here that we cannot find in Brazil. If you think you are, you can talk to me and then we can check, but I'm almost 100% sure that everybody can be found in Brazil. So I think this is a big opportunity for collaboration. And here is a project that has been financed already, and we are talking about heart diseases. So we are starting this project based on the distribution of heart diseases or heart-related diseases in Brazil. But you can see that it's very important also in Asia. And so I think there's a lot of opportunities for collaboration here too. And the idea here is that we're going to contribute to the HEART network that is also just starting. So we have a lot of discussions to do with them about protocols because we are about to start the project and we would like to start this project aligned with protocols that have been developed by this team. So I think it's also a very good following this map, opportunity to engage with Asia, to have samples collected in similar ways and understanding environmental issues related to this kind of diseases. What else do I have? So I brought some challenges. These challenges is for us to keep in mind when we are trying to design studies together. So I have a few slides on those. I think they are common to all the other Latin American countries, but also to Asian and African countries from the networks. So infrastructure and equipment access, I think it's a limitation for most of us. It was very interesting here in this meeting to see other options that we do not have in Latin America. So we have talked to them a lot to see what we could maybe showcase at home, back at home, as other possibilities for solutions. We have a lot of issues with high import taxes and logistics for bringing reagents back or to Latin America. I think this is common also to other regions. We do have funding constraints. Like I know when we show that we are like very well structured for funding, Vinicius showed the same about Chile, but the numbers are small. So that's why I showed you that we have to actually look at all the layers, and the partnerships are really important for us to be able to finance this kind of studies. Yeah, there is a mismatch also important between disease burden and funding allocation. So for neglected diseases, traditionally we have less funding. This has been like that, and it's continuously being like that. So I believe that if we partner with other countries that have similar issues, we can get much more far. Technical challenges that we have is we have been trying to help out with courses, capacity building locally. So this has been done continuously. We have to keep on doing it. We do not have enough training programs locally. Sometimes when we show the number of courses, it seems a lot for us, but it's really small. We know that it's really small compared to what happens around the world. We have limited access to training in general, data analysis capacity, which is also limited. And when we talk about sample access, there are also ethical bottlenecks related to transporting samples, collecting samples. And when we talk about sample collection, a lot has to be discussed. Ida talked a lot about this during this meeting, so how they had well-established protocols that they were able to communicate to the whole team. So afterwards, you're able to compare what you did. You have high quality of samples. We have a lot of biobanks locally, but not necessarily they have good quality samples for single-cell analysis. So a lot of this has to be discussed in the beginning of projects. Other challenges, we are generally underrepresented in global consortia, so we have to start to understand why. We have to participate more. We have to talk more about what we're doing to others so they can join. We do have barriers in publishing. This is important in high impact journals. We like to also sometimes put this there because we not always have money even to pay for the publications. There's the language barrier for many of us, institutional barriers. Anyway, and there are a few international partnerships when we think about it, especially with other areas of the world that look like us. So I think that we should invest more on this kind of partnerships with researchers that understand exactly what we're doing and the challenges that we face, and that we can together think about possible solutions. So takeaways from this talk: Brazil has high disease burden and low single-cell RNA-seq representation right now. So there is a clear research gap, and it's a big opportunity also for partnerships. National and international funders are accessible with the right collaborations and framing when we consider all the possibilities that we have for funding. We should have strategic partnerships with public health institutions in order to make these projects happen. We believe that, of course, single-cell RNA-seq studies can directly contribute to the public health system that we have in Brazil, which is like really widespread and lacks proper biomarkers and ways of looking at diseases. Investing in single-cell RNA-seq can provide precision insights for high-burden diseases, which we don't have right now, but also studies about the top causes of death locally. Those are more easily funded, and those are also common themes that everybody can discuss. And we believe the challenges should be understood and evaluated at the beginning of the study design, so we can have a successful project and we can, in the end, like really build from it. All right, so yep. I think this is what I have. Yeah, and this is our session tomorrow. We have a session on Asia and Latin America to discuss exactly these topics. And we will have researchers from Brazil and other Latin American countries, Colombia, Chile, Mexico, talking about what they're specifically doing in their single-cell research. That's it.
Thank you very much, Patricia. So before Marianne makes the next announcement, we have a time for a very quick question. Yeah, sure.
Patty, thanks for laying out all of the things that go into making these kinds of consortia. I'm curious about aging. There's a lot of buzz about single cell and aging. The problem is the aging phenotypes take a long time. So are you doing this in a cross-sectional way, or you have banked samples from long ago?
The two things, so there are biobanks, so this is what we're going to be evaluating about quality and what kind of research we can do with the biobanks. But it's yes, it's a prospective cohort. The idea, it's a 10-year project, and the cohort is based on people who work at this specific university, so you can really have the follow-up from these people yearly. Some of the sampling is going to happen twice a year.
That's amazing.
Please join us in this idea, right?
All right. Thank you, Patricia. Thank you. Maybe can you stand up here, because we are going to close the session soon, but we actually have 10 minutes just to have a wrap-up discussion. And I took notes of what to talk about, and Patricia just talked about all the items in the last 3 slides, which is a great talking points for how we can start thinking about and strategizing collaboration across globally, but as well as between Latin America and Asia and Africa and so on. And since we have a pretty large crowd here today, I think compared to tomorrow, I thought it would be a good opportunity to get some of your feedback or your comments or ideas and suggestions on how we can actually put this into an action item. And so I'm not going to do much talking, and I just really want to give this opportunity to you guys to just hear from you based on what Patricia just talked about, like some of the technology and budget issues and collaboration challenges. So anyone want to provide some comments and thoughts?
Yeah, hello, Vinicius again here. I believe as Patricia mentioned, to put together people, right? Because when we launched the Latin Cells Initiative, we never met in person before. We were discussing virtually because of the very first HCA Latam Symposium 2020.
During the pandemics, yeah, we couldn't.
Most of the people there never met in person. And then we started to have virtual meetings, and we managed to establish a team to apply for the CZI grant. And now I believe this transition to disease facilitates a lot, because many funding agencies, they only fund this kind of comparison. And we have a lot of synergies when we are talking about, for example, HIV. There are several. I remember last year we had a very nice talk with a group from Africa that they were generating a lot of single cell data in Africa from HIV. We are doing the same in Chile. All the tropical diseases like dengue, Zika, chikungunya, all the Asia and Latin America, not Chile, fortunately for Chile, but all the other countries in South America, we are affected by dengue, Zika, chikungunya. This is kind of project is something that we can try to do together, and taking advantage of this momentum that still people globally is thinking about diversity in genomic studies, right? Gallbladder cancer we have in Chile, but we know that we have high incidence in India and in other Asian countries. This kind of synergy we should try to focalize.
Like a tropical disease, so you have this tropical belt around the globe, right? And you have the South America, Africa, Asia that may share the same burdens, and that really will be another angle of looking at how we can collaborate across the world.
I believe that what maybe we need, because we all agreed that we need to do certain things, is to have the flagship project so people can really engage somehow. So maybe decide how, because I think HCA is a way to catalyze that, right? So maybe think about how this could be done so people could be joining, the idea.
Correct. And maybe we saw some technologies today where you can really scale, some like Ura-seq and others that can really reduce the costs significantly, and how we can distribute such technology around these places to also scale at low cost. So something we can also think about bringing the community. Sarah?
Yeah. So in addition to the kind of endemic infections, I was wondering whether vaccine programs are another kind of interesting way to have flagship projects across the world. And the, I know Fiocruz funds a lot of vaccine research, for instance, in Brazil, and connecting that, with a single cell, with the TCR, PCR, and so on, could be very interesting because you know exactly when the vaccination kind of hits, and so it's a sort of immune experiment in some sense. And I think so. We're working on some COVID vaccine, for instance, but it's a general principle that could be for any vaccine.
Yeah, I was
studies.
just yesterday talking to one Japanese researcher who is right there on this. He's an immunologist, and we were exactly talking about the fact that we have vaccination in Brazil that happens as a public system solution, right? So we know exactly when people are vaccinated, so we do have the time points, and we can do immunological research in that way.
So there's like immunization, as
program.
well as vaccination for prevention for the future. And perhaps some industries may be interested in this idea.
I mean, another place where the world can collaborate is octogenarian population. Start up with their PBMC and probably go with the skin fibroblast to see, irrespective of the genetic ancestry, if somebody is living 100 or beyond or close to that, there must be something in them. Probably we can use the single cell to start off with the PBMC, which may be relatively easy, and then go to the other part.
So you're saying aging, right?
Yes.
Yes, and PBMCs, absolutely.
Yeah, the idea of working with aging is because since we're going to have such a long cohort, like not all the results are going to be immediate, but that's why we want to work with the biobanks to see what kind of questions we can answer right away. And I think this is a very good opportunity to partner with other institutions who are interested.
And I believe groups in Thailand has also doing a lot of vaccines and dengue work and longitudinal follow-up studies. Any comments from the Thai team? Pompan.
So thank you. I discussed with Patricia yesterday, also discussed with Sarah, that it's possible to have some dengue vaccine, perhaps, that actually have multiple sites for clinical studies. And several countries, like in Thailand or in Brazil, the Qdenga is approved. So it's an opportunity. Maybe we can also work on that. And yesterday, during the infection and immunity and autoimmune talk, we also discussed beyond tropical infectious disease, dengue and also TB. There are people in the room that were quite interested. And also autoimmune disease would be another one that affect different population differently.
Yeah. Sham?
It's great to have this conversation. I think one question that always comes up is where's the funding going to come from? But I want to remind people that, from me, of course, I want to remind people that that is not the question you start with. Because by the time the funding call comes out, it's too late. If there's a request for applications, that request will be open for 2 months. You cannot put together this kind of consortium multinationally in 2 months. So it has to go the other way. You have to find like-minded people who are interested in the same thing, have samples that are relevant, want to study the same phenotype, and keep talking to them, brainstorming, so that when the funding proposal comes out, you have an idea. Or you already have a consortium, then you can start knocking on people's doors or from foundations or philanthropies or something like that.
Yeah, I completely agree. And with seed grants, I mentioned here some personal researcher grants in Chile, for example, where an individual researcher can start to generate some data that can be shared in the closed filter to build a consortium. And something very important at this very beginning is to share and discuss, because I believe this is important to discuss protocols, because to avoid different approaches, different tools, different methods, different kits, this is something that is, I believe, of key importance. So if you visualize something in the closed filter that could be a network, you can start from the very scratch, trying to focus a little bit more than normal in this kind of thing.
Thanks, Vinicius. I wanted to add to that on this question of protocols and so on, pilot data. So the other thing you need before the grant proposal is idea, you need collaboration, and then some pilot data, proof that you can share data, transport samples, and all that. Then you have a good grant proposal. And also, the pilot data helps you design a study that you think you can execute.
Cheyenne, can I just make a point about this sharing of stuff? Like, is the HCA hub for discussions maybe could be a way to make people connect? Maybe
John
wants to talk about that.
Yes, so this is a new membership platform that we just launched a couple of months ago, and it's intended to enable you to find each other, and to there's sort of discussion boards and so forth that you can start as well, job postings and so forth. So I encourage you to check that out and reach out to other members. The other thing I wanted to mention, particularly if you're putting together a consortium, feel free to reach out to the HCA Executive Office, because we have certainly in the past, in several cases, provided like a letter of support that connects the goal of your consortium back to the larger mission of HCA, and that's something that some consortia have found helpful.
I think this is a very good opportunity, no? That we could maybe have some threads.
So on that note, we have to close. I think the key is to connect, chat, and collaborate. And we need champions. We need someone who can say, hey, let's do this. And so maybe 4th C will be the champions, okay? So we need you to take the torch and let's bring people together. And I think that will be an excellent way to start the consortium together. So I was reminded that tomorrow we have day 4 meeting, which is going to be at a different venue. So it's not here, it's actually in what's called NTU at 1 North. So it's where A-Star, where Sham and I and a few of us are located, in Genome Institute is there. So please don't come here and go to this station called Buena Vista, and you'll be able to walk to NTU 1 North. And we're gonna continue this discussion, and we get to hear more about active science that's happening in Latin America, followed by also very cool science that's happening in HCA Asia. And it'll end by 12:50, and so they can have lunch and go home. All right, so thank you everyone for staying, and hope to see you guys tomorrow. Bye-bye.